Thank you. Thanks a lot for the invitation. It's really great to be here. Um, I put together um, since I figured that it would be a pretty diverse crowd. I put together something that is a bit more of an overview of the field that then goes deeper into my research interests, with the idea that maybe this would help spark interest from some people in the audience about current and possible research directions. But before going into the talk, I wanted to spend 1 minute talking about uh, this new university in Abu Dhabi called Mohammed bin Zayed University of Artificial Intelligence. It's a new university that's 5 years old, where myself and Michalis work. And we currently have six well-established programs, master's and PhD programs in different areas of artificial intelligence: natural language processing, computer science, computer vision, machine learning, robotics, and data science. And this year, we are opening computational biology. So a master's and a PhD program in computational biology, very broad, very related to everything that was discussed here today, but also a few other things. Um, and if you are currently on a master's, or perhaps if you're looking for a PhD or a postdoc, this is a really interesting place to be. Applications for the cohort open for next year will open in October. And I'm the program coordinator, so if you have any questions, just come talk to me. And it's worth noting that the student support is the best I've ever seen. If you count the housing support plus your stipend, it's about 4,500 US dollars per month, tax-free, which is a pretty good deal. And we currently have 27 faculty, but this is growing pretty rapidly on all different areas that you can think about, but very focused on health and translational initiatives, because the United Arab Emirates, they're very much about what can we apply towards healthcare. And so, and this is the last thing I'll say about the university. We are organized, each division is organized in terms of research groups that have their independence and individual research lines, but also a few core research directions that, this being a university of AI, are centered around what are the data resources that we can get access to develop new models. And we have three big projects in the division of biology and life sciences. One is a big partnership with a hospital, Cleveland Clinic Abu Dhabi, to create and bring AI to the hospital and to all the processes. The other is the Human Phenotype Project, which, if I have time at the end, I'll tell you a little bit more about, a deeply profiled cohort of several thousand people to create digital twins and to create predictive models of health. And the Emirati Genome Program, which is the world's largest genome program by numbers, with over 800,000 people. And so, to motivate my interest in machine learning and single-cell genomics, it's a common motivation for this crowd, is the interest in precision medicine. This idea that if we know what is going on at the molecular level, we will be able to tailor interventions to the point of the individual. But the challenge here, I don't know if I have a pointer or not, is linking the molecular and the phenotypic. And the really big challenge is, how are we going to link this at scale once we measure a lot of things? But even more than that, a challenge that we often don't talk about is, what are we going to measure? This is a cell. This is one of the smallest cells that we know of. This is actually a bacterium. It's not an eukaryote cell. And this is a simplified representation of the cell. So in yellow, you have the, yeah, this is not a pointer. In yellow, you have the DNA. In purple, the RNA. In blue, um, the proteins. In green, the lipids and the things that decorate them. And missing from here are the small molecules and other things. So when we talk about multimodal data integration in biology, this is what we mean. Can we put back together this picture from the data that we're measuring? Can we create meaningful models that let us understand what is actually going on from just like a very low resolution picture of the cell, because this is the best that we can do today. Even with single cell RNA sequencing, we're measuring a fraction of the transcripts of the cell and we're throwing away DNA, small molecules, protein. Can we figure it out? And the challenge, of course, is what are we going to measure? Because the cell has a lot of different chemical components, and some things are easier to measure than others. We talk a lot about DNA and RNA because those are easy to measure. We don't talk so much about metabolomics because we don't have good ways of measuring small molecules at a very large scale and a lot of molecules with single cell resolution. And I always like to anchor in the central dogma of molecular biology because that's how most of us tend to think of the cell. We tend to ignore all that other complexity, but also because the central dogma tells us a little bit about what we can actually measure. Because if we look at the arrows, information from DNA goes to RNA through transcription, but we have a way of turning RNA back to DNA with reverse transcriptase. But we don't have a way of doing that with protein. It's unidirectional, so it's talking about the flow of information. And because we can sequence DNA at high scale, we can also sequence RNA at high scale and generate a lot of data from RNA. With protein, it's much harder. And the big revolution that has happened in recent years, in the past 10 years, is that of single-cell genomics. Instead of taking a tissue that is made of many different cells and making a smoothie with that tissue, and then measuring the average of what's going on in the tissue and getting a vector and doing statistics on a few vectors, we now can measure the individual berries, the contents of the individual cells. And instead of a vector, we get a matrix. And then we do machine learning on that matrix. And we have been figuring out ways of making bigger and bigger matrices. This was the state of the field. When I started my PhD, every month, a new paper came out with a different method saying we did more cells. We came up with this new method. And if you look at the y-axis, it's a log scale. So someone would do 1,000 cells, 10,000 cells in one single experiment, 100,000 cells, 1 million cells in a single paper. And at this point in time, there were about 200 papers published in single cell. And when I was doing my PhD, we started a database to update this. And this rate of progress has not slowed down. 8 years on, we have about over 2,000 papers generating single cell data. And the volume of data has grown perhaps by 100-fold, the amount of data that we have available. It went from about 1 million cells to over 100 million cells. And this continues to grow. So now we have a matrix with 100 million rows that we can learn something from. Last year, in one go, a data set of 100 million cells was released by a company, Tahoe. And CZI announced that they want to do 1 billion cells. And now these kinds of scales are becoming commonplace. Many companies do routinely generate data sets with 50 million cells, 100 million cells. And BZI also has a project in this direction. And so this is a big challenge. How are we going to deal meaningfully with this volume of data? And something that a lot of people have been excited about is this idea of we have so much data, we should be able to apply these big machine learning models to create virtual cells, to create unified representations of these huge volumes of data. And the idea of a virtual cell is you have different data modalities, you create a good representation of each, you create a good predictive model of each, and you should be able to simulate the cell on every scale. There has been a number of larger teams pursuing these ideas because it takes a lot of engineering effort to organize so much data and to train such large models. One of them is based both at MBZUI and in the US, because it's a company that was born out of the university called GenBio. With this idea, let's create different foundation models for each scale, and let's, I do have a planner, and let's unify them. But they are not the only ones. A few other companies, including Tahoe, which is the company that released the 100 million cells, Zayed Therapeutics, the ARC Institute, have also done this. The challenge with these very large virtual cell models is that they are a big engineering effort. So they're not necessarily the best project for a PhD student. And if you look at what has been going on, most of what people have been calling virtual cells that are being published, they are often focused on perturbation prediction, meaning you want to find out what is going to happen to the counts in the gene count matrix if you add a drug to your cell culture, or if you knock out a certain gene. How is that going to affect it? And this is very useful, especially for drug development, and drug companies are pushing, driving a lot of investment in this virtual cell model, so it's understandable that perturbation prediction is one of the big focus. But it's not necessarily the main thing that most biologists in the lab would be interested in. And so I mentioned that these are very big engineering efforts, and I like to think about models in a spectrum. And on one end, we have analytically solvable models, like we actually just heard about, these analytical models for predicting the outcome of different diseases and of different treatments. And on the other end, we have very large models that are kind of black boxy, that require a lot of data to train well, but that we often don't glean a mechanistic understanding about what is driving the outcome. So they're able to make good predictions, but they don't give us mechanistic knobs. There's no mechanism in there. There's no understanding of the word. And in the middle, there's a number of different things that you could do that require a smaller team, smaller effort, less data, and less compute. And so generative models in biology can be used for perturbation prediction, but what about other tasks, and especially what about routine analysis, things that a biologist would want to do to analyze their data, their single cell data? It turns out that generative models in biology have not started being used just since foundation models became a thing. Sorry. But before I explain you that, I want to point out that we've had not only an exponential increase in the data that we're generating, but also an exponential increase in the tools that we have made. There has been almost a one-to-one correspondence in like a paper being published for a single cell data set and some custom analysis package being done. In the past, this was almost 1-to-1. Now it's decreasing. But when you have so many tools, it is also difficult to even know what to do, to even know what modeling approach to use. And this is for the bioinformatician or for the biologist in the lab that is generating some data, what their analytical landscape looks like, right? You went to the lab, you did an experiment, you want to analyze it to get some results, to know, to learn some biology. This is kind of what you have to do oftentimes. And what is important about this picture is that the steps shown here, they all rely on more or less standard machine learning and methods. There is nothing out of the box. And those are fairly well established, but they are not unified necessarily under a consistent statistical framework. And what the usage of generative models in biology started enabling was the possibility of bringing these under a coherent framework. So I mentioned that generative models in biology are not a new thing. Since at least 2017, they began being used to model single-cell genomics data. Not these huge foundation models, but small models based on variational autoencoders that turned out to work pretty well to both model the data, to do to create a good latent representation, to do clustering, to do cell typing, and to do different kinds of analysis. And the thing that made these models work well with small amounts of data, like 10,000 cells, was the fact that they captured something about how the data was being generated in the system, meaning what was the biophysical process by which the data was being generated, whether something about the cell size, something about how the sequencing process happens, something about how the assay itself generates the data. And I'll give you two examples to illustrate. But before that, I need to tell you a little bit about how the data is generated. So most single-cell genomics approaches, they rely on, most single-cell genomics data actually, relies on microfluidics. So in order to sequence the contents of individual cells, you need to somehow isolate them physically and then tag the RNA of the cell with a barcode that enables you to identify where that cell came from. And you can either do that in plates or wells, or you can do that in a microfluidic droplet. And in order for you to put your DNA in that droplet, you use a little bead. So the big ball is the bead, and the small ball is the cell that is coated with DNA. And this DNA will pair with the, will capture the RNA, and then you will do some more chemistry, and you get your gene count matrix. And if we're interested in modeling a little bit of this process, we can think of the cell as a bag of RNA and protein and the other things that I showed you on the first picture. But for our purposes, we only care about the messenger RNA, actually, which has a polyadenylation tail, which will then be captured by the bead. And we will do reverse transcription and then PCR. We will amplify the RNA, and we will end up with a tube that is full of copies of the original transcripts of the cell. And each color here represents an original molecule. So a molecule may end up with 4 copies, 5 copies, 100 copies. And of course, you know from which molecule it came from and from which cell it came from. And so I drew this picture here because the process of sequencing, generating this data, actually is the process of sampling an urn of colored balls. But it's actually a tube of tagged transcripts, a tube of barcoded transcripts. And this is the generative process of your gene count matrix. If you look a little bit into the biochemical steps that make this data. And so that's why we this is a matrix of counts of natural numbers, 1, 2, 3, very few, very sparse. That's why we need to use some probability distribution to model count data if we're doing this. But there's other parts of the process that we could equally think about in these terms. And I illustrated all of this to you to show how in one of the first VAs used for single cell, it comes together. So I drew to you the cartoon of the cell, and here is the generative model of the cell. So for each cell that we have, it has a certain cell size. It has a certain latent representation. It comes from a certain batch, a certain lab, which we know who generated the data. And then each gene has a certain activity and a certain number of gene counts, which are being sampled to generate the gene count that we see in the matrix. And then there's some noise, dropout, because we see a lot of zeros, and that's modeled here. But in this generative model, actually, when we're fitting to the data, the only neural networks that we're training, I think I lost my battery, are 2 small neural networks of 1 or 2 layers, depending on how you configure your model. And so this trains very fast, in 1 hour, 2 hours on a GPU with very, very little data, 10,000 cells, for example. And this is just to illustrate the idea of how you can bring a little bit of your understanding of the system to your model. Another kind of data modality that we have is site-seq, which pairs measuring the RNA with measuring the protein. And the way that we're able to measure both is that we have some antibodies that bind to certain proteins on the surface of the cell, and they have this little tail of poly A. And so they pretend to be an RNA, and they're captured just the same. And so the generative process would be the same, except for the fact that the antibodies are often not very specific, so they bind to other things. So you also would have to model that. And you can do that. Another extension of SCVI called TotalVI has been published that accounts for this non-specific bind. And this illustrates the idea that if you want to model a certain kind of data with small generative models, you can. And that this principle, this idea then, right, our idea of can be extended to multi-omics data, to what are fundamentally just many different matrices of data that may be paired or not. And this approach worked well. It's been turned into different models for different data modalities. And a consistent challenge was, how do you link the machine learning frameworks, PyTorch, with the single cell analysis tools that we have. And this eventually coalesced as part of SCVI into this larger framework called SCVI tools that aims to make this the development of these models accessible, meaning easy to use for biologists, but also extensible. So if you develop a model, it can live under this framework. And the model here is the model architecture, not a foundation model. There's other repositories for that. And if you want to try a different model on the same data set, you just change 1 line of code. This framework is SCVI tools is part of a larger community of Python-based tools. But what is really nice is that it standardizes development of these small models in a way that is both easy for the user and easier for the developer, because you only have to worry about what is it that you're implementing. And these are small models. These are some of the models that are currently part of this zoo of different model architectures, and for many different kinds of data modalities. And this is some of the extensive documentation to help um, the user. So if you publish a model, you'll get the paper tied to the model in the framework, and you can write the documentation for your model. And this all lives under the SCVerse community of Python tools for single cell analysis. I'm going to skip this just to wrap up with this idea that the point of this talk was to illustrate this spectrum of modeling options that we have, and this idea that variational encoders, generative Bayesian modeling, graphical models can offer a way for us to embed a little bit of what we know about how the data is being made in a way that makes the models both more interpretable, but also faster to train, more lightweight, less data hungry, and easier to use. And this is my closing reflection, that if we have enough data, big models do tend to win. But because there's so many different things that we can measure in biology, we often don't have enough data for the new and interesting things. And if we don't have enough data, we need to make more or less ad hoc models. But now we are at a place where not a lot of data is still enough data for us to do machine learning, just not to train a huge foundation model, but it's still a good amount of data to make a principled model that is manageable by one person or by a small team to make some biological discoveries. So with that, thank you very much.
Thank you, Eduardo. So now for questions. Does anybody have any questions?
Thank you, first of all, for a wonderful presentation. I actually have a question about the single cell sequencing data. Because I've seen some paper publications doing deliberately bulk data analysis to bulk data analysis to stabilize and to denoise the information in the single cell RNA sequencing. How reliable, in your own perspective, is the single cell RNA sequencing data, and is bulk still necessary in this case?
Yeah, so if we look at the gene count matrix, like here, essentially what we will have after we do the cell typing is we identify what are the different types of cells present. And most often what you're interested in is what is the difference between two cell populations. So you want to compare the red cells with the blue cells. It turns out that if you're very good at labeling your cells, and then you pseudobulk, meaning you sum up just the cells that are red and just the cells that are blue, you get a very good profile of that cell population. And then you can actually use methods for differential expression, for example, that have been developed for bulk omics, and they work very well. You can get very good single cell profiles and with the pseudobulk with as little as 20 cells, if you have 20 cells that you sequence reasonably. But if you have 50 cells, you have a good profile. And so this tends to work very well. And the reason is that these bulk methods have been extremely validated. They're extremely robust and based on well-established statistics. There's not a lot of new methods. But also because we still leave a lot of the power of single cell data on the table. We don't really have hierarchical models that often take so much advantage of the structure of the single cell data. So for many analysis steps, in particular for differential expression, pseudobulking works very well.
Thank you very much.